I think we can get started. How about questions related to lab today? Anything you'd like clarification on? As long as we're somewhat fresh in our brains, having had lab just an hour ago or so. Any questions on lab?
Professor, this is Haley. Did you still want me to talk to you after class?
Probably. I'm trying to remember what we were going to talk about. I know you weren't in class today.
I have COVID.
Oh, okay.
Yeah.
Yeah, so we should talk afterwards.
Okay.
Yeah. Any stuff on lab? Okay. Obviously, there's not going to be many questions on chapter 2 chemistry because we're done with that for forever and ever. According to the schedule, you should be into chapter 4 by now, or at least starting it this week. So I would think if I were you, my goal would be to get through, half of it by the end of this week. And then next week, we've got it listed as well. So we got like 2 weeks for chapter 4. And chapter 4 covers just a ton of information, a broad variety of different topics, some of which are not easy. So I would anticipate probably lots of questions about chapter 4. But we can talk about 1 or 3 as well. Anything you'd like to review? Like me to go over? Things that aren't making sense?
I have a chapter 4 question, but I just wanted to give everybody a chance to ask about the previous chapters, in case they want to review.
Sure. We can do whatever, Alexis. We can talk about chapter 4, or we can review chapter 3 material. I mean, you tell me what you would like to do. Everybody's aware that a week from today is our first lecture exam. And as long as we're talking about that, maybe we should talk about how much of chapter 4 should we put on the test. So give me a second here. I want to check something out. Yeah, all right. Look at the syllabus here real quick. Well, here's sort of the dilemma we have. If we don't put all of 4 on test 1, then test 2 is going to have 1, 2, 3, 4, 4.5 chapters on it. Ordinarily, I don't like to not give you adequate time, to go over material before a test. So that's why I was thinking of putting, part of 4 on the second exam, which we can still do. But then that's going to mean you're going to have at least 4.5 chapters. That's a lot of chapters.
So are you saying we have the option of doing all of chapter 4?
Yeah.
In the quiz if we want? Okay.
Well, I'm just wondering, as I think more about it and look at the schedule,
Right.
even though chapter 4 is a bit of a challenging chapter, it's not terribly long. As I look at it here, it's starts on page, 122. That's the cover of the chapter. And then it's done, it wraps up on page 145. So it's actually only about 20 pages. It's not really that long a chapter. It covers such a broad variety of topics. Cellular respiration, DNA replication, protein synthesis. These are pretty broad, important topics. I mean, I guess, and I'm not saying this to stress people out or whatever, but I think it might be worth, in the long run, putting all of 4 on the test next Wednesday.
Yeah, I'd be happy with that.
I
'Cause
agree.
if we don't, then your chapter 2 or exam 4 is going to be a beast of a lot of chapters.
I'm in for that if everyone else is.
Yeah.
I mean, I don't know what your prerequisites are for this class, but I know that when I had biology, we already did all of this, so it should be review.
Paige, you're absolutely right. It should be review. And you've all heard of this at some point in your high school biology, if not even a college bio course. So you are absolutely correct. This is nothing new. It's just that the topics are, again, they're just typically challenging for students. Which is all the more reason to make sure we do a review before Wednesday. So I'm going to make that happen. We talked a little bit about it in lab, but I will find an hour somewhere, probably Sunday or Monday for us to get together and talk about this. Yeah, we'll make that happen. So
Not including the separate one that Kira does, right?
yeah, this would just be something I would say, hey guys, if you're interested, I'll make myself available. I'm just gonna, I'll pick a time and a date off the top of my head. I'm not promising this, but Sunday at 2:00 in the afternoon or something.
Okay.
Or Monday. I have to check with the boss, like I said, to see what her thoughts are.
Right.
Yeah.
Okay.
Yeah. But
Okay.
I will send an email out. So be looking for an email throughout the weekend, and we'll do that. So I'm going to just suggest that we do put 4 on. Okay. I hope you've had a chance to get into 4. If you haven't, you should have. You can't be falling behind. You got to keep adhering to the schedule as best you can, the lecture schedule, So what would you like to talk about? Are there specific sections? Again, I'm assuming you had a chance to look some of this over. This is our agreement, right? You go online, you look at the Zoom lecture, and then when we get together, we can get into more detail or we can review specific stuff. If there are no questions, then I can just pick something and start talking about it, but I would rather have you give me some guidance. But if you don't have any questions, then I guess I'll just start somewhere.
I had a question about the catabolism and anabolism.
Yep.
Just because there's this one paragraph and I feel like it just, because I know this isn't like a biology class, so I think they're trying to like skim over it, but it's on page 125 in chapter 4.
Yeah.
And it just says that both catabolism and anabolism must be carefully controlled so that the breakdown or energy releasing reactions occur at rates adjusted to the requirements of the building up or energy utilizing reactions. Any disturbances in this balance is likely to damage or kill cells. So I just kind of wanted to talk more about, that balance on, how quickly they release energy and stuff like that.
Yeah, that's a great question. So let me share the PowerPoint. Okay, everybody see that?
Mhm.
Okay. So what page is referring to is a paragraph in the catabolism section there in 125, like she said. And so what you need to understand, of course, is what are these two terms, anabolism and catabolism. And I talk about that in the PowerPoints, and there are slides here that I'm showing you that review that. So the first one here describes the mechanism by which bigger, more complex molecules are made from smaller, simpler ones. That's called anabolism, the buildup of bigger molecules from smaller ones. And this process, which occurs in cells 24/7, 365, is an energy requiring process. So for example, in this particular slide, we're looking at how two monosaccharides are joined in a chemical reaction referred to as dehydration synthesis. Synthesis meaning to build or make. And you can see what's the end product, the bigger, more complex disaccharide. And notice there's a water produced. So a couple things here. Where does this water come from? Do you see where the water comes from? If you go back into the reactants, look at this blue highlighted area. That's where the water comes from. So an OH group is pulled off this monosaccharide, and a hydrogen is pulled off this hydroxide group of the adjacent monosaccharide, and a covalent bond forms between the two. This oxygen that I'm pointing to with the arrow is this oxygen right there. Okay. In other words, have we accounted for all of the elements, all of the hydrogens and the carbons and the oxygens here in the products? The answer is yes, but we got to remember there's two hydrogens and an oxygen that were part of the reactants, but are now forming a separate water molecule in addition to the disaccharide. So in this reaction, in this anabolic reaction called dehydration synthesis, where the cell is making a bigger, more complex molecule, it's making a water molecule as well when those are joined. And getting back to Paige's question, this requires energy for a cell to do this. It doesn't come free. So there are times when your cells need to generate bigger organic molecules, like enzymes, let's say. You know, enzymes are important for digestion, aren't they? Well, where do you think those enzymes come from? It's not CVS or Rite Aid. It comes from your cells. Your cells make all those dozens and dozens and dozens of enzymes to help you digest your food, which you then absorb. So that's an energy cost that's built into the whole system, if you will. The other part of metabolism, the other half of it, is called catabolism. And this is just the opposite of anabolism. Here, a cell is going to take a bigger, more complex molecule and degrade it into smaller, simpler ones. So we, in essence, have reversed the reaction, haven't we, from the previous example, where now we're taking, the cell is taking this disaccharide, incorporating water into the reaction, and in a hydrolytic reaction, it's called hydrolysis, right? The end result is the formation of two simpler sugars. And notice that this water that is part of the reaction gets incorporated back into the products here, isn't it? Yeah, so again, it's just the reverse. Notice that this reaction, breaking things down into smaller, simpler forms, releases energy. Energy release. So there are times when a cell might need to generate heat, like to maintain body temperature, for example. Or, well, that's probably the best example I can think of. To help maintain your body temperature at a constant 37 Celsius or 98.6 Fahrenheit, your cells need to break down food. That's why you eat, to make energy. Yeah, because if you run out of energy, What happens to your cells? Well, Paige, you alluded to that. It says, quote unquote, any disturbance is likely to damage or kill cells. So if you, A, run out of energy, your cells die because they can't keep the body temperature at 37. They can't pump against the concentration gradient. Remember, that's active transport. Remember us describing that in one of the earlier chapters? This was the movement of materials from a low to high concentration, right? 40% of a cell's energy usage goes into active transport mechanisms. So in essence, Paige, what that gets at is the body, the cells of the body are, in an effort to maintain homeostasis, are balancing the need to make energy with the needs to burn energy, if you will, or use it. So there's a balancing act that's going on all the time. You are taking in food so that you can undergo anabolic reactions. That takes energy in the form of ATP. This is the energy currency of the cell. But just as quickly as we are, making these bigger organic molecules, whether they're carbohydrates or lipids or proteins, we also are breaking them down to make energy. So it really boils down to an energy usage sort of thing when you get right down to the physics of the process. It's kind of interesting. It gets really involved, but it's so
Okay.
much philosophical in that sense. Go ahead.
And is that why our bodies kind of store fat? Because if we just had too much energy, it would make us, so they have to do something with it. Is that kind of that process?
Well, our body makes fat when, we're supplying the cells with adequate sugars, let's say, and those are being broken down adequately to make enough energy to supply our needs. And so any excess sugar, we either excrete it in our urine, for example, or poop it out, which we don't do really either. We conserve that, and through evolutionary processes, we take that extra sugar and we store it as fat, as you say. Why do we store it as fat? Well, because the day may come when we run out of the ability to take in food, and we can rely on those energy stores. Now, in today's society, it doesn't work that way. But in going back 300,000 years, when we roamed the plains of Africa, there weren't very many fat, Homo sapiens running around because there wasn't excess food. But if there was extra food to be had, then we would store it as fat because someday we would want to tap into that energy to sustain life. It's really an evolutionary throwback thing. Because you might remember from chapter 2, fat contains twice as much stored energy as carbohydrates or proteins. It's a very effective form, fat is, to store energy. And again, you can just think of an animal like a deer, puts on a lot of fat in the summer and the fall because now it's slim pickings. I had a doe on Sunday as I'm eating breakfast, walk right by the window in the middle of the day. It was like 10:30 in the morning. And you don't see deer typically out in the middle of the day. They're always hiding in the woods, right? Well, it's become evidently clear to me that deer are getting pretty hungry, and they're getting more brazen in terms of appearing in the middle of the day eating my shrubs. I mean, I like to see them. I like to eat them too, to be quite honest, but I love to see them. They're beautiful animals. But when you butcher a deer, if you will, or process a deer in the fall, it's amazing how much fat they put on, and that's to get them through the lean times, right? Not like right now in the middle of winter. Now, another thing that I want to mention about these two reactions, or processes, I should say, is the fact that enzymes are also used both in the synthesis of bigger molecules from smaller ones and in the degradation of bigger molecules into smaller ones. I don't really say much about that on these two slides, but your book goes into a nice description, actually in the next section, where it talks about control of metabolic reactions and the importance of enzymes. Boy, enzymes are just absolutely critical They are important. I don't know if that answered your question or not, but yeah. How about other questions? Chapter 4 stuff. Do you know what enzymes are? Do you know how they work? Really important. By now you should be into that.
Can you go over how they work?
Sure. So enzymes are catalysts. Catalysts help speed up reactions. What kinds of reactions? Well, catabolic and anabolic reactions, like we just talked about. Hydrolysis and dehydration synthesis. Those are reactions that involve the degradation or buildup of molecules. And without enzymes, our cells would have a hard time living. It would be too costly for them to undergo anabolism and catabolism. So we have these proteins called enzymes to help speed reactions up. Another interesting thing about enzymes is that they're reusable. And these enzymes are very specific with regard to what substrates they will help to convert into products. And that's sort of what this slide talks about. The other thing to make note of is that enzymes are proteins. So on the left, we're going to depict this enzyme, this purple shaded enzyme, as having a particular shape or conformation. It's the term that some books use, conformation. It's a fancy word for the shape of the molecule. Now, you remember me talking in lab today using my microscope cover as a cell, and we talked about how when you cut it, you might not catch all the chromosomes and all that stuff. Okay, so you know that a cell is three-dimensional. At least you should know that. You don't think of it that maybe as a three-dimensional entity, but it is. Cells are three-dimensional. They're tiny, they're microscopic, but they have height, width, length, all that stuff. Would you be surprised if I told you that molecules also have a unique three-dimensional conformation? But you never thought of that very much. Well, if I asked you, have you ever seen a DNA model? Either, up close and personal or on a nature TV science show. How many have seen a DNA molecule? Model of a DNA molecule? Yeah. Some of you are shaking your head. Others of you are like, what is he talking about? So if you did see it, can you describe it to somebody who's never seen one? What would you say? How would you describe it to somebody who's blind, let's say? DNA.
I would tell them it looks like a spiral staircase. I don't.
I agree with you 100%. That is a great, great metaphor to use. Absolutely perfect.
Like a
It is like,
together.
I'm sorry?
Like two ringlets wrapped around each, like intertwined.
Okay.
Ringlets of hair, kind of, I guess. I always pictured it like that.
What if I told you, picture a ladder that has been twisted?
Okay.
Well, that's like a spiral staircase, same thing, right? Okay, so it's indeed what a typical model of DNA looks like. It looks like a spiral staircase. The steps in the middle of the spiral staircase or the ladder, okay, think of the ladder if you want. The steps of the ladder, do you know what those indicate or represent? What kind of bonds? We talked about it yesterday.
They're hydrogen bonds.
All right, hydrogen bonds between the nitrogenous bases, exactly. So back to my previous statement. DNA has a three-dimensional conformation or shape. So do all organic molecules, lipids, carbohydrates, proteins, and nucleic acids. So to sort of try to depict the fact that enzymes, being proteins, have a three-dimensional conformation, we're going to think of those molecules as having a particular shape. And so we're focusing on a particular region of the enzyme called the active site. Okay. So the active site of the enzyme determines what substrate molecules or molecule will fit into the enzyme, like a lock and key analogy. Okay, same idea. In other words, can you use your car key to open up the lock of your house or apartment? Obviously not. The key to your car only fits into the car. Well, in the same general way, this particular lock, if you will only accept a certain shaped key. Now, please understand this is a metaphor. This is not like real. I'm not saying you have locks and keys running around in your bloodstream, all right? You know that. So here's two substrate molecules. Maybe these are two monosaccharides that need to be combined to make a disaccharide. Good example of that. So these substrate molecules fit into the active site of this enzyme because their shape conforms to it, right? Everybody see that? Yeah. So when this process of the enzyme combining with the substrate occurs, we form what's called an enzyme substrate complex. So here you see them all complex together. And then later, within a very short period of time, like milliseconds probably, the product is produced. And here is our unaltered enzyme that can be reused if there's more substrate to combine to make more product. And there's a YouTube video that you should watch that talks more about that. But notice that these arrows are reversible, indicating that these enzymes can also sometimes be used in reverse processes, if you will, to take the product and break it down into its subsequent building blocks. So if we go from left to right, that's sort of anabolic, isn't it? That's dehydration synthesis. And if we go from right to left, what is it? It's catabolism. It's breaking down bigger molecules into smaller ones, right? So we're just applying what we learned here with the action of enzymes. So they're extremely important, these enzymes are. And as I said a moment ago, they're not just involving buildup of bigger molecules from smaller ones. They're also involved in breaking down bigger molecules into smaller ones. In fact, I would say most of you, when you think of enzymes, you think of their role in the digestive system. Most people do. Is that anabolic or catabolic? Digestion.
Catabolic.
Right, it's catabolic. But I'm here to tell you that enzymes also work in anabolic processes as well. So here we've got an example of a substrate that's going to be converted into a product. We're going to call it substrate number 2, and you'll see why in just a minute, but it's basically being converted into some other substance by a particular enzyme that we will call enzyme A. So its active site accommodates the substrate, and the end result is the formation of a different molecule, right, and a reusable enzyme, just like we saw earlier. Look what's happening next. That substrate number 2 can be converted into another substance called substrate 3 by a different enzyme. And substrate 3 can be converted into another substrate or substance through the action of a different enzyme. And finally, in our example here, enzyme D is going to be instrumental in converting the substrate, number 4, into some desired end product that the cell wants to make. So in order to make this end product, the cell had to utilize how many different enzymes? 4 of them, right? Yeah. 4 different enzymes were involved between the beginning of our substrate 1 until we made the desired end product. What they're trying to get you to think about is the fact that it's not just one enzyme converting one substance into a product. It often involves a series of dozens and dozens and dozens of individual enzymatically driven reactions before a cell can produce a desired end product of some sort. Now, how does a cell know? Because it's not really thinking, right? It's not a cognizant entity, it's a cell. But how does the body regulate how much end product to make? Does it make sense to make more than you need generally? No. And so the book talks about this process that involves this first enzyme called a rate limiting enzyme, excuse me. So in many metabolic pathways, or in some they say here, the product inhibits the first enzyme in the process to ensure that not too much product is produced. And I say in the slide here, this type of control is an example of chemical negative feedback. Now, don't confuse this with physiologic negative feedback. In physiologic negative feedback, we talked about that in chapter 1. For example, how do you maintain constant body temperature? Right, that's physiologic. We're talking here about how does a cell know to make enough product? And if it makes too much of it, what do you want to do? Well, you want to slow down or reduce the concentration so you don't make too much. That would be inefficient. But you want to make enough for the needs of the cell or the needs of the body. So what this is saying is that initially these various substrates are converted by these different enzymes to make some desired end product. When the desired end product level gets to be optimal, and we don't want to make any more of it, the concentration, the increased concentration of the product has an inhibitory influence on the first enzyme such that this enzyme A begins to convert less substrate 1 into substrate 2. If you convert less substrate 1 into substrate 2, what's going to happen to the levels of substrate 3 and substrate 4? It's going to go down. What's going to happen to the level of the product? It's going to go down too, right? Okay. Now, what if the product level gets below where it should be? Then what do we want to do here? Do we want to continue to inhibit enzyme A? No, we want to inhibit the inhibition. A double negative is a positive. We want to promote the rate limiting enzyme if the product level gets too low. By promoting it, what's going to happen to the product level eventually? Hello? Am I going too fast, or have I lost you? Are you all sleeping? Are you all watching TV? What's going on?
I was writing it down. Can you just say the question again?
Okay.
A little too fast, that's all. Sorry, sir.
Okay, no problem. If the product level, no, I'm just kidding. Yeah. If the product level drops below where it should be, what do we want to do to that product level? Negative feedback says we want to bring it up. Right?
Yeah.
If it gets too low, let's bring it up. If it gets too high, what do we want to do? Bring it down. Right. That's what we're talking about with respect to negative feedback. So how do we take a high level of product and lower it? Well, we cause the first enzyme here to stop working or to slow down its conversion of 1 into 2. If it converts less 1 into 2, you're going to have less 3 produced, less 4 produced, less product produced, and it'll drop. Okay, so back to my question. What if the product level gets too low? Well, you're all going to say, we want to bring it up, right? Yeah, duh. Well, how do you bring it up? By
Either one
promoting, not inhibiting, but promoting enzyme A. Like saying, enzyme A, please convert 1 to 2, right?
Right.
Now, notice, are we talking at all about enzyme B, C, or D? No, we're talking about this guy, which is why I circled it and put it in pretty colors to draw your eye there. You have no idea how much time I put into these PowerPoints. You guys have no idea.
Well, I was just going to ask you if you could show it again. Yeah, they really are helpful. I'm sure you did.
The real key is understanding that this whole mechanism hinges on what enzyme A does. Yeah. So again, getting back to Paige's earlier comment or question, a cell, a tissue, an organ system, an organism has these built-in mechanisms. It's amazing. It's like you don't have to think about it. It just happens. It's part of your physiology and your biochemistry. It's just remarkable to make sure that you're running at top form. You're this highly regulated, well-evolved thing that just does amazing things. You know, it's a fine-tuned process. Metabolism, not to get into a lot of detail, but I mean, it sounds like a boring topic, right? Metabolism, oh my God, right? Well, when you start to study it, you really, you start to think about it more, and it's like, wow, this is pretty amazing. Okay, so there's a quick comment on enzymes and how they work.
Okay, and on our study objective kind of list there,
Yep.
it says explain how metabolic pathways are regulated. Is that just that chemical negative feedback that we just talked about?
Yes.
Okay.
Yes, that's what I was getting at there. Yep.
Okay, cool.
Yep. Yep. The next slide here that talks about ATP is extremely important as well. And I think you saw in one of the earlier slides when we talked about energy usage or energy production, you saw this this 3-letter term ATP. And so here we define what that refers to, adenosine triphosphate. This is the energy currency of our cells. This is the form energy takes on. Now, you know that the mitochondrion is often affectionately referred to as the powerhouse of the cell. That's how you learned it in high school. That's how I learned it in high school. It hasn't changed. So this organelle is the important organelle of your cells that makes this ATP stuff. Yeah. Because if you run out of it, as I say here, the cell dies. And if the cell dies, what happens to the tissue? Because tissues are made of cells. You know that from chapter 1.
Right.
The tissue dies. If the tissue dies, what happens to the organ?
Dies.
Right. Organ system dies.
Right.
Organism
Just one
dies.
after another.
It all builds. Right. So you are composed of trillions of cells, all of which are making energy. Because without energy, you would die. You need energy to maintain body temperature, to pump materials against a concentration gradient. Your muscle cells need energy to contract, right? I mean, it's just critical. You run out of energy, you die. And so this ATP molecule is really, really, really important. And what's going on as we speak right now is our cells are generating ATP, our mitochondria are, because our body cells need it. But what are we also doing? If we're using ATP, we're using energy. Do we have an unlimited supply of it? It's like taking a battery and in your let's just say in your cell phone, and then you use your cell phone for 3 days. You use the energy in the cell phone, and then of course you suddenly look at it and you notice what? It's down to 10%.
Yep.
And you plug it in. We all do it every day or two, right? So how do we plug in our cells? How do we regenerate that ATP? Because we know we need it. We got to be able to regenerate it. And that's what the next topic is in section 4.4. It's called cellular respiration.
So can I just, is this where like the electron transport train and, okay.
That's
All
right.
right. Okay.
That's part of it. That's right.
I just wanted to make sure I was thinking.
Yep, you
Okay.
are. You are. Yep. That's a part of this process that you see on the screen next called cellular respiration. This describes basically how our cells generate ATP. Yep. And it's basically made up of several different steps. In essence, what cellular respiration helps to do is it helps to phosphorylate ADP. Well, okay, what is that? Well, here's adenosine triphosphate that was defined in one of the earlier slides. See where it gets the name triphosphate, the 3 phosphate groups?
Mhm.
This little wavy line means unstable. It's technically a covalent bond, but it's an unstable, a particularly unstable covalent bond. And that ATP, when our cells use it, is degraded into ADP and phosphate. Let me go back to that slide. Now, where is it here? Right here. So here's the breakdown of ATP into ADP. That helps cells do whatever cells need to do. Contract. and
Did
so
you
on.
pull it up? It's not on our screen.
Not
Or
on
we can't, no, I didn't know if you were sharing or not. I thought so.
Yeah, I thought I was. Okay, hang on.
I didn't mean to interrupt.
No, I'm glad you did, because I thought you were looking at it. Sorry. Okay, so there we go. Now do you see it? Okay, thank you. Yeah, always interrupt me if you're not seeing something. I'm sorry about that. Okay, so here's the cycle. I was saying just a moment ago that our cells are always using ATP for contraction, for moving materials against a concentration gradient, whatever the case may be, right? Without energy, we die. And so when our cells degrade ATP, we basically follow the right-hand side of this cycle, if you will. ATP is degraded into ADP, adenosine diphosphate, and then this phosphate group that's pulled off, the terminal phosphate is called, this ends up bonding to another substance, which results in things like muscle contraction or pumping the material against the concentration gradient or whatever the work might be. So there's this phosphate group, it's called. Phosphorylation is the attachment of this phosphate group onto some other molecule, which results in work, cellular work. But we just said a moment ago that we need to recharge the battery, right? You got to plug your cell phone in. So we've got to resynthesize this. How do we resynthesize ATP? Well, we basically take the ADP and we phosphorylate it to make ATP. So we're basically just shuttling around phosphate groups. But in order to phosphorylate ADP, we have to undergo this process called cellular respiration. This is what's going on in our mitochondrion, our organelle, the mitochondrion. This is the powerhouse doing the work. It's phosphorylating ADP to make ATP. And this mechanism is called cellular respiration, as I said a moment ago. And it's made up of 3 basic steps. And I've outlined those in purple and blue and yellow. Now, this particular slide, as it says here, an overview, is giving you a broad overview, if you will, a broad perspective of the mechanism. And then the next few slides, we go into more detail of what's happening in those three stages or steps, whatever you want to call them. Do you guys remember studying this in biology? Sadie, you never had this in biology?
So when I took biology, my teacher got pregnant and she never came back. So
What?
we had,
That's no excuse.
no, so then we had a substitute that like a regular substitute so we didn't have a teacher for like 2 weeks, and then they gave us another substitute that had like kind of a science degree, and then she didn't know what she was talking about. And then right at the end of the year, Mr. Leiter from Allegheny retired, and he came to Franklinville then and then taught biology for like the last 2 months. So my bio record is not very good.
You go to the school board and complain.
It's already over with.
See, I feel really bad because if this is the first time you've heard of this, it's like, shoot me now, right? Because this is hard stuff. I feel bad.
Yeah, I don't remember this either.
Well, you probably don't remember it because it was so painful at the time. You just put it out of your mind.
That's me. I just blacked it out.
Yeah. That's because at the time, you know, the teacher was pushing this stuff and you all were like rolling your eyes in your heads like, oh my God, what does this have to do with my life? How am I ever going to use this? And little did you know that on February 10th, 2021, it was going to come back to haunt you. Okay. So what this describes are the 3 basic steps, okay, of aerobic. It's called aerobic respiration. Aerobic means you need oxygen for this to occur. In glycolysis, let's look at the word. What does that literally mean? Dissect the word, might help you figure out what's going on.
We don't have a screen again. Are we supposed to?
Well, I just switched slides. Why did it, maybe I went back to none. Okay, maybe that was my fault. So there's the slide. Here's the first of the 3 stages or steps. What is this word? If we dissect it, I think you might be able to figure it out because you heard of hemolysis today in lab. What does lysis mean? Lysis. When a cell lyses, it bursts, right? Like the red cells. When placed into a hypotonic solution, they popped, remember? So this is the breakage of glucose. That's what that refers to, glucose. So let's look at glycolysis in the next slide. Everybody see that? Okay. This process occurs in the cytoplasm of our cells. And as it says here on the slide, this occurs in the absence or presence of oxygen. Now, earlier you heard me say that respiration is aerobic. And it is. Just bear with me here a little bit. But technically, the first step of cellular respiration, glycolysis, will take place whether you have O2 present or not. Now, most of the time, there's plenty of oxygen getting to our cells because our respiratory and cardiovascular systems keep up with the needs of our cells. Okay? 99% of the time, we can supply adequate oxygen to the cells and this process occurs, but it can occur without oxygen also. It's actually a 10 different chemical reactions going on here in glycolysis. We're not going to go over all 10. You don't need to know all 10. You should just understand kind of what do we start with and what do we end with, and what do we make along the way? Okay. What do we start with? What do we end with? What do we make along the way? We start with glucose. We end with 2 3-carbon compounds called pyruvic acid. So for now, ignore the stuff down below. Just look at where I'm circling with my arrow. Notice the number of carbons here. 3 times 2 is 6. That's how many we had in the original glucose, right? So we have to be able to account for everything, and we can. But between the beginning molecule of glucose and the ending molecules of pyruvic acid, there are different things made along the route, if you will. In the cytoplasm now, this is taking place. Well, what do we make along the way? Well, you'll notice here that there are 2 ATPs degraded into 2 ADPs. There's also 2 phosphate groups that are pulled off, one from each ATP, right? Remember that from the earlier ATP cycle? Remember that? When ATP is degraded, you make ADP plus a phosphate group. Okay, here are the 2 phosphate groups that came off these 2 ATPs. So this molecule has been phosphorylated. That's the word they use, phosphorylated. And then this compound undergoes some additional splitting and breakdown. And toward the end of glycolysis, look what happens here. 4 ADP are phosphorylated to make 4 ATP. So we break down 2 at the beginning to get the ball rolling, if you will, and we make 4 here at the end. So what's the net gain? 2, right? Minus 2 plus 4 equals plus 2. Everybody good with that? Calc 3? Okay. Okay, so we are coming out ahead, a net gain of 2 ATPs thus far. Remember, the whole goal here is to talk about how much energy can a cell make. That's the whole purpose we're talking about this. For every glucose that a cell starts with, how many ATP molecules can be produced? So we've thus far produced 4, but we've also broken down 2 at the beginning, so we come out ahead a net gain of 2 thus far. In addition to the ATPs being made here toward the end of glycolysis, you see here 2 NAD plus gets converted into 2 NADHs plus some hydrogen ions. And in the Zoom recording of chapter 4, I talk about what NAD plus acts to do. It's basically an electron carrier. Okay. And I liken it to an empty school bus. This is something I made up. You're not going to see this in the book. I'm going to write a book of my own. I'm going to make $1 million someday. So here's the empty school buses. There's 2 of them here. They get passengers in the form of electrons. Here's our electrons. They get on the empty bus, and we make NADH. John, that looks like you. Is that you on the bus? No. All right, now you're going to see why NADH is important in a few minutes. But it's all important to gather electrons, because those electrons are going to be used in what we call electron transport chain, and that's where we're going to make a lot of energy, a lot of ATP. So far, we've come out ahead a net gain of 2, but these NADHs are going to help us make a lot of energy later. Okay, and they can do that due to the passengers they're picking up in the form of electrons. So 2 empty school buses become 2 full school buses here. Okay, any questions on that? Okay, I am recording this, so you can go back and watch this, again, if you want. let's play this video. I don't know if you're going to be able to hear it or not.
Cells derive energy from the oxidation of nutrients such as glucose.
Can you hear it? Very lightly. Okay. All right. I can't turn up my speakers, but you can play this yourself. But I'll play it anyway. But
Cells derive energy from oxidation of nutrients such as glucose. The oxidation of glucose to pyruvate occurs through a series of steps called glycolysis. The energy released during these oxidation reactions is used to form adenosine triphosphate, ATP, the energy currency of the cell. The initial steps in glycolysis are the additions of 2 phosphates to the glucose molecule at the expense of 2 molecules of ATP. The result is a 6-carbon sugar diphosphate molecule and 2 low-energy adenosine diphosphate molecules, or ADP. This 6-carbon sugar diphosphate molecule is then split into 2 3-carbon molecules. Each of the 3-carbon molecules is converted through a series of steps to pyruvate. During these reactions, electrons are transferred to the coenzyme NAD+ to form NADH, and ATP is formed. Under aerobic conditions, the pyruvate is further oxidized to yield more ATP, and under anaerobic conditions, the pyruvate is converted into lactic acid.
so this pyruvate that the video talked about, that's the same thing as pyruvic acid. Don't let that confuse you. It's the same molecule. And at the end of that video, they said that under aerobic conditions, the pyruvate then enters into steps 2 and 3 of aerobic respiration, which we'll talk about next. If there is no oxygen, okay, because we said this could occur in the presence or absence of oxygen, right? If there's no oxygen, then the pyruvate is converted into lactic acid, and this is what can accumulate in your muscles and cause muscle cramps. Okay. Lactic acid, maybe you've heard of it before.
And then, so is that, so lactic, and then that occurs just until like the oxygen gets there? Like, what's it, oxygen deficit? Does that go with this too? I'm just trying, I feel like I'm remembering this. So,
Yes,
okay,
yes,
good.
yes,
Okay, good.
yes.
Okay.
Yeah, you build up what's called an oxygen debt, and that has to be repaid. You got to convert this lactic acid back into pyruvate or pyruvic acid.
Oh, okay.
Your liver does that. We'll talk about that in the muscular system chapter. Yeah.
Oh, okay.
So this NAD, empty school bus, that holds the electrons, this is called a coenzyme, NADH, and that's what the video mentioned, coenzyme. It's an enzyme helper. But for now, just think of it as an electron carrier. Okay, this is the empty bus, NAD with a plus superscript, and the NADH is the full school bus, loaded with electrons. So now we enter the step number 2 of aerobic respiration. You can't do this unless you have adequate oxygen present, and I've noted that here on the left-hand side. Now we transition from the cytoplasm into the center of the mitochondrion, called the matrix region. Okay, and again, you can go back to chapter 3 of the book, where they talk about the mitochondrion, and I think they define the matrix and the folded inner membrane, which is referred to as the cristae. Okay, so to review, you got the innermost region of the mitochondrion called the matrix, and then you have these infoldings of the inner mitochondrial membrane called cristae. It increases the surface area. We'll talk about why that's important in a few minutes. You have the outer membrane of the mitochondrion here in orange, and then the light orange region, this is called the intermembrane space. Inter means between. It's between the outer and the inner mitochondrial membranes. So you should be familiar with this anatomy of the organelle. It's important. So in the citric acid cycle, occurs within the matrix, the center of the mitochondrion. We're going to follow what happens to one pyruvic acid. Remember, there's 2 made in glycolysis. So one of these pyruvic acid or pyruvate molecules comes into the mitochondria, go into the matrix. And there we pick up another busload of electrons. So another NADH is made, big bus of electrons. We also pull off a carbon. There it goes. Carbon dioxide is released, and a 2-carbon compound is produced. You don't need to know the name of it, but it does have a name, acetic acid. And that acetic acid basically combines with a 4-carbon compound that was made at the end of the preceding cycle. Okay, the citric acid or Krebs cycle is basically what's going on here in the center of our organelle. So this was made at the end of an earlier cycle. It combines with the 2-carbon fragment of the pyruvic acid, right, that lost a carbon. And we now produce a 6-carbon compound called citric acid. And these gray spheres are just simply representing the carbons. There's also hydrogens and oxygens and other things coming off of this. This is just the skeleton of the molecule. But that's where the name citric acid comes in. It's the first compound produced in the cycle. This 6-carbon compound gets converted into another 6-carbon compound. And you go through like 1, 2, 3, 4, 5, 6, 7 different reactions until you come back to a 4-carbon that can combine with another 2 carbons coming off another pyruvic acid, and you just keep cranking through the system. There's inputs coming in that combine and you crank through. So what do we make as we go from citric acid all the way around the circle back up to the 4-carbon finish molecule? Well, we make some more NADHs. You see 3 of those here, right? And you see 1 ATP being made directly. And then you see this molecule here in the lower left called FADH2. FADH2 started its life as FAD, which you can think of as the small empty shuttle bus, just like you could think of NAD plus as the empty school bus. This is a bus, but it can't carry quite as many passengers. So it's a shuttle bus. When that fills with passengers like we have here, those passengers being electrons, we form FADH2. This is another coenzyme. So for every cycle through this, the cell is generating, an NADH here at the beginning, and then 3 in the citric acid cycle, 1 FADH2 and 1 ATP is being made directly. So if I were to ask you, how many NADH molecules are made per original glucose in the citric acid cycle, what would your answer be? How many NADH molecules are made in the citric acid cycle for every original glucose?
4? Or is it just in the actual cycle, so we don't count the top?
Yeah, don't count this top one because
This
it's
is
technically not
3.
part of the cycle.
It's 3.
So there's 3 for every revolution, but I said per glucose. How many pyruvic acids are made per glucose?
Oh, 2.
2. So 2 times 3 is 6. Yeah, right? The question was, how many NADHs are produced in the citric acid cycle per original glucose? So you have to know that the glucose yields 2 pyruvic acids, and each of those enters the cycle. Does that make sense? Okay. So we have gathered up a lot of big school buses, haven't we? We've made, are you still seeing the PowerPoint? Okay, we've made 2 here, right? And then we've made 3, 4, 5, 6, if we count this guy up here, 7, 8, 9, 10, because we got to go through this twice. So we've made 10 NADHs per original glucose, 10 big school buses. How many shuttle buses have we made per glucose?
2.
2, right. So I want you to picture in your mind, you know, we've got all these big school buses and a couple of small school buses heading down I-86, and they're traveling toward the inner mitochondrial membrane now of the organelle. I'm not going to play this video. You can watch that. You should also
When
watch.
glucose is oxidized during
You should also watch this video. This explains what happens to those electrons that we've been storing in the big school buses and in the smaller school buses. Okay, because we've been gathering these up, haven't we? Now, what we have right here is the mitochondrion, right? You all recognize it, I hope. What's the orange? What's that called? The center? Matrix. Matrix. What are these things here called? They're extensions of the inner mitochondrial membrane, but we gave them a special name, started with a C.
Kitels?
Cristae. These are the cristae. And what did I say about their shape? Help to do what?
Add more surface area.
Right. You can have a lot more surface area here of the inner mitochondrial membrane, shown here in blue, can't you? By having those cristae invaginations. That's important because it's here in the inner mitochondrial membrane where a lot of this energy is going to be made. So we're going to zoom in and I will play this video. Just sit back and watch this. Come on.
When glucose is oxidized during glycolysis and the Krebs cycle, the coenzymes NAD+ and FAD are reduced to NADH plus H+ and FADH2. In the mitochondria, the electrons from NADH plus H+ are transferred to the electron carrier proteins, and the protons are transferred across the membrane. As the electrons move from cytochrome to cytochrome down the electron transport chain, more protons are carried across the membrane. Cytochrome C transfers electrons to the cytochrome C oxidase complex. Protons are also transferred to the outside of the membrane by the cytochrome C oxidase complex. The cytochrome oxidase complex then transfers electrons from cytochrome C to oxygen, the terminal electron acceptor, and water is formed as the product. The transfer of protons generates a proton motive force across the membrane of the mitochondrion. Since membranes are impermeable to ions, the protons that re-enter the matrix pass through special proton channel proteins called ATP synthase. The energy derived from the movement of these protons is used to synthesize ATP from ADP and phosphate. Formation of ATP by this mechanism is referred to as oxidative phosphorylation.
Okay, so let's watch that again without the volume on. So what this is talking about now is what happens to those electrons, those passengers in our buses, right? They play a really important role. That's why it's called electron transport chain. So NADH is going to be giving up its electrons and become NAD+, the empty school bus. And that's happening now. So there's the empty school bus, NAD+. Guess where it goes? Goes back into glycolysis and the citric acid cycle to pick up more passengers. Okay, it's a shuttle. That's all it is. This is the empty bus, NAD plus. This is the full bus. Where is it? Full bus. NAD plus is the empty bus. NADH is the big full school bus. This is the full shuttle bus. So those electrons are pulled off, and they're going to get passed from protein to protein to protein within the inner mitochondrial membrane. This pink and purple and blue, these are proteins. This is the inner mitochondrial membrane. What does this resemble? It should resemble something. Phospholipid bilayer, embedded proteins. Where have you heard that before?
The cell membrane.
Right, the cell membrane. So what I'm basically telling you is that whether we're talking about the cell membrane or many of these organelles like the mitochondria, they are sharing very similar chemistry. So these proteins, they acquire the electrons that have been given off by the NADH and the FADH2, and they're passed through special enzyme proteins. You don't need to know the name Q, C, the oxidase. You don't need to know the names. Just kind of watch the process. Now, as those electrons are passed from protein to protein to protein, you see these hydrogen ions moving from the matrix into the intermembrane space. There they go. And there's an accumulation of these hydrogen ions here in the intermembrane space as a result of these electrons getting passed from protein to protein to protein. So we pump a lot of hydrogen ions through the inner mitochondrial membrane out into the space. Now you might say, what happens to all these electrons? Well, eventually the electrons, as you're going to see next, they're going to combine with oxygen and some more hydrogen ions to make water. So water is actually an end product of electron transport. But more importantly, how is a cell making energy here? That's the whole idea here. We're trying to see how a cell can make a lot of energy from glucose. So those hydrogen ions are now going to move from this intermembrane space where they've been accumulating in high concentration. See them all accumulating here? They can go back in, but only if they pass through a special protein called ATP synthase. It's a special membrane protein. And as they pass into the matrix, look what happens to the ADP. It becomes phosphorylated to make our high energy ATP. And that's really an important takeaway. So for every NADH that gives off its electrons, the cell will generate on average about 3 ATP molecules. And for each FADH2, the smaller shuttle bus, for the electrons that it gives off, and the associated hydrogen ions that accumulate in the inner membrane space, when they come back in through the ATP synthase into the matrix, the cell generates about 2 ATPs. And so my question that I posed to you in the Zoom lecture is, how many ATP molecules can a cell generate aerobically for every glucose that it starts with when it goes through all of these different steps? Well, I give you the math here, and you should look at that and work through it. And when we get together this weekend or Monday, you can ask questions. How about any specific questions as we wrap up class? Yeah, this is not easy. You got to sit down, you got to study it, you got to look at the PowerPoints, slides.
So like on the exam, will we be able to see this, or like will you just ask, and so like we should just like embed it in our brain that like there's 6 made after like the cystic cycle, or like is there going to be a diagram?
There probably will not be a diagram.
Okay.
So you want to try to picture it in your mind's eye.
Thank you.
Do you think most questions will just be on certain aspects? Like we won't be asked to like, notate the whole entire cycle, right?
No.
Okay.
No, I'm not going to ask you to explain the electron transport chain. No, but you should understand basically what it's there for. What does it do? It's here again that the cell generates the maximum number of ATP molecules. So again, I think what you should ask yourself is, do I know what do I start with? What do I end with? And what do I make along the way? That's the key thing you should be focused on with regard to glycolysis, the citric acid cycle, and electron transport. What do I start with? What do I end with? What do I make along the way? Okay, well, you've got a lot to ruminate over. Again, watch the Zoom lecture. I talk about all this. Watch it a couple times if you need to. And I'll be in touch with you, and we'll see if we can't get together some day, either, I don't know what day it'll be. Look for an email. I'll send an email out. Try to give you 24 hours advance notice. Okay, we'll meet on the Zoom, in the Zoom room like this. And again, as you're studying this, write things down. Write down questions. Say, check on this. It's not making sense. Okay.
Can I have the question on the chapter 3?
Say that again, my day.
I have the question on chapter 3.
Okay. I have to run to an appointment soon because we're at 2:30 now. But I can hang around for a few minutes.
Okay.
If you'd like.
Yep.
Okay. So basically, everybody else, I will see you sometime this weekend. How's that? Okay.
Yep. I confused between the area concentration. How you know what is the higher area of concentration and the lower concentration?
Ask that question one more time. I didn't catch the first part of it.
Yeah, so when we study about the diffusion,
Yeah.
and we talk about the area have the high concentration and the low concentration, how you know that where is higher and where is lower?
Yeah, oh yeah, okay. So can you give me an example of what you're talking about? Is there a diagram in the book?
So it's about the osmosis.
Yeah, are you looking at your book?
Yeah.
What figure? What figure? What number figure?
No, on the PowerPoint page 14.
Okay. Let me pull it up. Is this the slide you're referring to? Yes? Okay. Wait till my buddy goes by here. Okay, so your question is, how do we know which side is the higher concentration?
Yes.
Okay, well, it depends on what you're talking. Are you talking about the concentration of the water or of the solute?
Oh, it will be different.
Well, the solute is depicted by these little pink, purple and blue dots, right?
Yep.
So in this particular example, what this is referring to is the fact that you have more solutes here on part A of the beaker than you do in part B of the beaker. Okay, so I guess the blue dots they're depicting here is water. All right, so they're not solutes. The blue dots are the water. The purple dots are the solutes. Well, they're calling them proteins, same thing, solute. So where is there more protein, on A or B?
A.
There's more concentration of protein on this side, right? So water is going to move from a higher water, lower solute into a lower water, higher solute concentration. There's still some protein over here, right? You see the purple over here, but there's more concentration of them in A. So water is going to move across, just like saying there's more molasses over here than here. Water is going to move toward the higher molasses. And so the effect will be over time that the level of water in A rises and the level of water in B falls. Now, you'd never see that happen in the lab, but this is theoretical.
Oh, okay. Yeah.
So when you ask, how do we know where the higher concentration is? You got to ask yourself, are you talking about concentration of water or concentration of solute? And they define, again, the water as the blue, smaller dots, spheres, whatever you want to call them. And the purple, the bigger purple, would be the protein solutes.
Oh, okay. So I know, like, we have two different protein, water concentration and then the solid concentration, right? It's not like one area higher and another will be lower.
Well, in this particular setup, there's a higher solute concentration on the left side here in A than there is in B. And because there's a higher solute over on this side, water will always move toward a higher solute in an attempt to reach equilibrium, where there's an even distribution of solute and water. So if you look now, relatively speaking, the purple and the blue ratio is about the same on both. There's more protein here, but there's also more water. The relative spreading out, if you will, of the solutes in A is about the same as B. So we've reached what we would call equilibrium. We couldn't do that unless the water moved over to the left. I don't know if I'm answering your question or not.
Yeah, I kind of confused between to figure out what's the water concentration and the solute.
I'm going to have to tell you, here's the water, it's blue. Here's the solute, it's purple. I would tell you that. You wouldn't just assume it. I'd have to tell you which is the solute, which is the water. Or like today in lab, I didn't color the water molecules. I just put the solutes in. Those X's were solute molecules. The rest was water.
Oh, yeah.
So as long as you know where the solute concentration is, you just got to remember water always moves toward a higher solute.
Mm-hmm.
Does that help?
Yeah.
Okay.
I think I will watch this video again and
yeah, go back into chapter 3 Zoom lecture. There should be more, my description is there, and there may even be an animation. I can't remember if I put an animation in there or not.
Yep.
Okay, great.
Thank you.
Thanks. Freya, did you have a question? You're muted.
Yep. No, I'm good. She was pretty much covering it. I didn't know if, yeah, I'm okay now.
Okay,
Okay, thank you.
sure. Because I have to talk to Haley a little bit here. Okay, so Freya, can you leave the room so I can chat with Haley? And Maivei, can you also leave the room, please?
Yep, thank you. Bye bye.
Bye-bye, thank you. Okay, Haley, are you there?
Yep.
Did you say yes?
Yes, I'm sorry.
Okay, so what do we need to do? You missed lab today.
Yep, and that should be the only class I miss. I'm out of quarantine on Sunday.
Uh-huh.
And then I'll get my rapid test done Monday morning at work to make sure that I'm not positive. I just need to make up the chemistry exam from today.
Okay. I don't have that in electronic form to send you.
Okay, I can come in whenever. I mean,
Okay. But the problem is I'm not going to be in for the rest of this week, and Monday and Tuesday we don't have classes.
okay.
So I think what we're going to have to do is unfortunately have you take it next Wednesday, along with when we have our first lecture exam.
Okay.
I don't know any other way
That's
around
fine
it.
with me.
Yeah.
Yeah, it's kind of an inconvenient time to get COVID.
Right. Did you test positive for it?
Yes, I did.
And how are your symptoms?
I mean, I have a different symptom every day. I completely lost my taste and smell. Like I'm just not doing too hot.
Uh-huh. Yeah. Well, but you're not like going to the hospital.
No, nope. I can still breathe and everything. It's just
Yeah.
rough.
Yeah. Okay. Well, that's good. So I think, well, what we said in class today was, since I don't know, are you in the review or not?
Yes. Yep.
Okay. So, the students asked if I would do a review session, like on Monday or Tuesday. And I said, I can't do it Tuesday because I have faculty development that day. I have meetings all day. And I said, we have a cabin in Allegheny this weekend, but that I would try to come home on, Sunday or Monday, and I can do an hour Zoom. So you might see an email, you will see an email from me indicating a time when I can do an hour kind of review.
Okay.
There's also a tutor. I think I've told you guys, Kyra Nolder. She's doing a Sunday evening Zoom tutoring. I just wanted to let you know about that or remind you of that.
Yeah.
Okay.
Yep.
Okay, great. So then next Wednesday, we have our lecture exam scheduled, and we're going to be doing exercise 8 in lab.
Okay.
Yeah. So you're going to have quite a bit to do when you get back here on Wednesday.
Yeah.
Now, if you want, you can hang out after lab and take the chemistry exam. I can give it to you, at when lab is finished. And just outside my office here, I've got a couple chairs. You can take it in the hallway.
Yeah, that might work better so I don't miss anything within the actual lab. Yeah, I can do that because I'll just hang out and stay for lecture, like go to the library or something.
Okay. Yeah,
Yep.
that's great. So
All right. Perfect.
I'm going to put you down on my calendar here.
Okay.
So chem exam, I'll just say at noon.
Okay.
It's a 30 multiple choice questions. It'll probably take you literally 20 minutes or so to take. So you'll be done by 1, no question.
All right. Perfect. Thank you so much.
Yeah, sure. And then as far as the lab today goes, we spent some time looking at some slides. This was in exercise 7 of the
Okay.
cell cycle.
Uh-huh.
The lab was open all day Wednesday. I mean, you're welcome to come and stay and, do whatever,
Mm-hmm.
but you should look at those, at a couple slides of the cell cycle, because we will have the lab practical the following Wednesday, and that's going to be on it, as will exercises 5 and 6.
Okay.
Exercise 5 has a cell model in the lab that the students need to look at, and there's a handout for that in Blackboard.
Okay.
So you should definitely look at the cell model. You should review the organelles. It's a fairly short exercise 5 in the lab book. It's really small.
Okay.
And then exercise 6, I can't re-demonstrate what we did, unfortunately. Do you know anybody in class you can get the notes from?
Yes, I do. I have a couple people in my math class today, actually. So
Oh,
I'll
well,
just reach out to them.
that would be great. And then if you have any questions, you can certainly talk to me about that on Wednesday too.
Okay. Yep.
Okay, great.
Alrighty. That sounds like a plan.
Okay, well, I hope you're on the mend. It sounds like you're on.
Yeah. I'm kind of just recovering.
Yeah. Are you glad you're not at school or working?
I'm actually more stressed out now that I'm not at school or working.
Yeah, I know.
I'm ready to be better, like back and
Yeah. But I'm saying, if you could go back to school or work, do you think physically it would be a big issue, or you could probably get through the day?
I think I'm a CNA, so I'm pretty
Ah.
sure that would be an issue. But
You just hear people who get it, and they're just so down, physically just exhausted, they could never function.
yeah, it wasn't horrible, and I don't want to like downplay how bad it is for other people, but
Yeah,
it definitely wasn't fun.
Sure. Do you know how you got it?
My best friend tested positive, and she's like one of those like really big anti-maskers, all that. So she was around me, and I was pretty mad at her. I was like, I work with the elderly. Like I can't.
Yeah.
So I told her, I was like, listen, if I test positive, I'm gonna come beat you up.
Yeah.
But we get tested 3 times a week at my job, the rapids. So I mean, as soon as I tested positive, I knew.
Yeah.
So
Yeah. Well, I'm glad you're handling it okay. That's good.
well, thank you, and thank you for letting me make this up.
Oh yeah, absolutely. I'm sorry that you ended up missing the chemistry exam and the one quiz.
Yeah.
But missing the one quiz isn't the end of the world. But we'll get you that chem exam makeup next week.
Okay, perfect. Thank you so much.
You're welcome. If there's any questions, just reach out to me too.
All right, thank you.
Okay, bye, Haley.
Bye.
Bye.